So continuing on with our discussion on the nature of the interaction between the spike protein and the virus viral membrane. Right. So again, the spike protein is made of actually three separate protein subunits. All right. So it has what's considered quaternary structure. All right. So the first thing that I want to discuss about this is why is it, what is it that contributes to allowing a protein to hold its shape? All right. If, as we said before, each protein is essentially a chain of amino acids. All right. Well, when I think of a chain, I typically think of a straight line. All right. But this thing is definitely folded around on itself, and it looks like it has a particular shape. All right. And that shape is gonna, it's what eventually makes these little red blobs here. All right, and the shape is extremely important. All right, for one thing, the outside surface of it right here, that's what makes up the binding region. It's the specific shape and the specific amino acids that are at the tip of this that create that specific binding interaction between the virus and its host cell. All right, so these proteins. This protein being able to maintain this particular shape is very important for that virus, because if it didn't have that, there's no way it could have that interaction with the cells. So the key thing to remember here is that this thing is interacting both with other amino acids, but it's also interacting with its environment. All right, and one of the key things to remember is that each one of the amino acids in this chain has what's considered an R group. All right, so in that chain of amino acids, each one of those amino acids has an R group, and the thing to remember is that some of those R groups are going to be polar, and some of these R groups are going to be nonpolar. All right, so for example, alanine, the R group is a simple methyl group. All right, that would be considered a nonpolar R group. All right. But the R group here, arginine, it's got these carbon-nitrogen bonds, so there's more polarity there. Asparagine is going to be considerably more polar. It's got this carbonyl group and these nitrogen-hydrogen bonds. All right. So depending on the side group, for example, cysteine, that's going to have a non-polar R group. All right. That side group, that R group, is either going to be polar or non-polar. And the reason that's important for protein shape is, let's just take a simple example of let's pretend that this protein is just floating in an aqueous environment. All right. Well, if this is floating in an aqueous environment, how would it hold its shape? Well, the inside of this protein, if you were to be in the middle of this, that's the part of the protein that's not exposed to the water environment. So those are going to be your the inside parts of this are going to primarily be the molecules that are either that are nonpolar. All right. There's another way. But if there are groups in there that are polar, they're going to be two polar things that are essentially right across from each other. All right, because they could stick to each other. All right, and that would help to hold its shape. The outside of the protein here, remember, that's going to be where the water environment, if this thing were to be floating in water, would be primarily your more polar groups. All right, so things like that arginine. Okay, so things like aspartic acid, that would typically want to be something that's on the outside, because this carboxylic acid could hydrogen bond with the aqueous environment out there. Whereas cysteine right here, this would typically want to be something that's on the inside of that, sticking to other nonpolar things. And that's a fairly simplified way of explaining how this thing would hold its shape if this thing were to be a protein that's floating in water. All right, but remember, this S protein is actually embedded into the membrane of the virus. All right, so that's let's take a look at that. So here's a graphic showing a protein that's embedded into a cellular membrane. So again, here you have your phospholipid bilayer. All right, the gray areas are where you have that the phosphate head group. All right, and remember that head group is got is polar in nature, and so it really likes the water environment out here. All right, on this side, you've got a polar head group, and it likes the water environment of pretend that this is the inside of the virus. All right. Alternatively, this could be the inside of one of the host cells. All right, but either way, it's going to be an aqueous environment. All right, but then you've got the long chain fatty acids that make up the lipid bilayer. And so remember, this part right here really doesn't like polar groups. All right, what this shows right here is this is a protein which is embedded into that lipid bilayer. All right, and one important thing to remember is that this protein, there's no covalent bond between this lipid bilayer and this protein. In fact, there's no covalent bonds between this phospholipid molecule right here and this phospholipid molecule right here. All right, they're just held together by London dispersion forces. All right, nor is this phospholipid bound to this phospholipid right here. It's just that, again, London dispersion forces create areas of stickiness here. All right, it would actually be energetically unfavorable for one of these things to float off, because if one of these things floats off, all of a sudden this nonpolar group right here has got to come into contact with water, and it really doesn't like that. All right, so how does this work? All right, well, as we mentioned before, you've got different R groups on your that make up the R group of your amino acids on your protein. All right, and so if you take a look right here, notice that all of the R groups that were on the amino acids right here would be in contact with those long chain fatty acids. So this region around here, like, remember, we have a choice. I mean, like, proteins don't think, but if you had to make a prediction, what type of amino acid side chains would make sense to exist in this portion of the molecule? Would it make more sense to have nonpolar ones like cysteine or alanine, or let's find another nonpolar side chain? Methionine would be nonpolar. Proline would be nonpolar. So would it make more sense for those areas to be nonpolar, or to have polar side chains, things like glutamine or glutamic acid? All right. Well, since those amino acids are in this part right here, which are right next to a nonpolar environment, all of the amino acids here are going to tend to have nonpolar amino acids, things like alanine. All right, on the outside there. All right, same thing on this side of the molecule right here. All right, but notice that there's parts of the protein that extend outward. All right, and so in the coronavirus, so that explains the part of the protein that's going to extend down into the phospholipid bilayer. All right, so the parts of the spike protein that you can't see because they're embedded into this gray area right here, you would expect those R groups would be very nonpolar. All right, and outside here, you're gonna you also have R groups that are exposed, but notice this is going to be an aqueous environment out here. All right, so the R groups that you would expect there are going to be your more polar R groups. Things like glutamic acid, aspartic acid. Oops. So this illustration right here just pretty much sums things up. The parts of the protein that are outside of the membrane tend to be your hydrophilic or water-loving side chains. All right, again, those are the things that are going to be able to tend to want to be able to make hydrogen bonds with the water molecules out there. The parts that are within the membrane are going to tend to be hydrophobic. All right, again, there's no covalent bonding that holds these things in. It's just that it would actually take energy to pull this out. All right, one way to think about it is if you had put a drop of oil onto water. All right, it's actually more energetically favorable for the oil molecules to stick together in a ball than it is for them to spread out and become part of the water. All right, and it's not because there's a covalent bond making those oil molecules stick to each other. It's just the nature of polar bonds like other polar bonds and nonpolar bonds like other nonpolar bonds. So that's it for today. Next week we're going to head into some other aspects. We'll eventually be moving on to the topic of DNA and RNA, because inside of the virus, and one of the main things that the virus wants to do is it wants to get inside of a host cell, and then insert its genome and use the machinery of that host cell to replicate itself. So we'll take more of a look at that in next week.